Okay, AP Bio, are we ready to dive into cellular respiration? Because that's what we're doing today. We're going to talk about the details of this whole process. You'll want two things in front of you today, your Unit 6 notes, of course, but also your diagram of a mitochondria. Take a minute and make sure that you have labeled that and colored that and you know where these different parts are, because we're going to be talking a lot about these as we go through today. So you have this diagram printed as a full page in your note packet. We're going to come back to this several times throughout class today. This is the roadmap for the process of cellular respiration. If we go through it from the beginning, here's the glucose that we start with. It goes through three main steps. It goes through this process called glycolysis, which you can see from this picture happens outside of the mitochondria, just in the cytosol of the cell. That produces some ATP and also some of this molecule called NADH. Remember, anywhere you see hydrogen, hydrogens carry electrons that carry energy. So this will be an important molecule. The pyruvate that's made through glycolysis enters the mitochondria and goes into this process called the citric acid cycle inside the mitochondria that makes some more ATP, also produces some of these molecules, NADH and FADH2. You can see both of these have those hydrogens that carry electrons that carry energy. And that brings us to the third step, which is the electron transport chain. There's a lot of vocab. There are several processes that happen here. This produces a lot of ATP. So this is kind of our roadmap. We're actually going to be working our way backwards through this, but we're going to continually refer back to this diagram to remember where we're going. First, though, I want to talk a little bit about this NADH and FADH2, these molecules that you see coming out of these first couple of steps. NADH and FADH2 are coenzymes. That means they help enzymes to function, and both of them has these variable forms. You can see they can exist. NADH exists as NAD+. FAD also can exist without those hydrogens, but then you can add hydrogens to those molecules to create this NADH and this FADH2. So these are kind of, I think of these sometimes kind of like buses that shuttle electrons attached to hydrogens back and forth in this process. These are the full buses. These are empty buses that can take more hydrogens. The sciencey way to say that is that in this form, they are electron acceptors. This is the reduced state. They have received those electrons attached to hydrogen and can transport them to the next step. So their job is to shuttle electrons from the first two steps to the electron transport chain. So that we'll come back to that. But the electron transport chain, remember, the reason we're interested in those hydrogens is that they have a lot of potential energy. Electrons attached to hydrogen have potential energy because they can easily be pulled off that hydrogen and kind of fall down this electronegativity hill that we talked about on Friday and release energy as they do so. So this idea is going to be important as we think about electrons moving. Remember, as electrons move off of hydrogen to different elements, they are releasing energy that can be used for other processes. So here we are. We're going to start at the end of our map. Our end goal here is to make ATP. There's a little bit of ATP made in these first two steps, but the majority of ATP made in cellular respiration comes out of this last step. So we're going to start by looking at how ATP is actually made. That happens with the help of this molecule. This is a complex of proteins called ATP synthase. And as you can see from this diagram, it is located in a membrane. You can see a lipid bilayer right here. But if you notice the labels, this side is the mitochondrial matrix. And this is the intermembrane space of the mitochondria, which means this protein complex, this purple thing, is actually located in this membrane right here. That inner mitochondrial membrane, that is where ATP is actually made, between the intermembrane space and the matrix, that middle part of the mitochondria. And of course, there isn't just one of these. There would be ATP synthesis found in several places throughout this inner mitochondrial membrane. And the reason that inner membrane is so folded and convoluted and has all those cristae is to provide lots and lots of surface area, so there's plenty of room for all of these proteins. If our goal is to make ATP in this process, the more surface area we have for this protein complex, the better, the more ATP we'll be able to produce. And the way that this works, you can see there is a very high concentration of hydrogen ions in the intermembrane space. So this space in here between the outer mitochondrial membrane and the inner mitochondrial membrane has lots and lots of protons in it. Another way to say that, you can say it as a high concentration of protons. You can also say that it has a low pH, because remember, pH measures the concentration of hydrogen ions. So I just want to mention that because sometimes they will refer to it that way in test questions. So just remember, a lot of hydrogen ions gives that a lower pH. And there's a lower concentration of those in the mitochondrial matrix. So hydrogen, of course, is going to want to flow down its gradient. That's called the proton motive force. This is really a fancy way just to say that the protons want to move down their concentration gradient, and that's a source of potential energy. The only way that the protons in this intermembrane space can diffuse across this membrane and get into the matrix is through ATP synthase. This protein complex creates a channel that allows proteins to travel through it. And so that's the only way that they can get in. And as they do that, hydrogen is a positively charged ion. And of course, a positive charge is going to interact with all the R groups that make up all these proteins and cause them to change shape. And so the movement of those hydrogen ions actually causes this protein to move in such a way that it takes adenosine diphosphate and those phosphate ions and joins them together to create ATP. This is a little ATP making machine that is powered by the flow of hydrogen ions down their concentration gradient. So that's what happens at the very end of this process. That's how we actually make ATP, is by the diffusion of hydrogen through ATP synthase. So ATP synthase makes the ATP. Where do we get that proton gradient? Because if we need protons to diffuse, that means we need to create a difference in concentration. Otherwise, it would just reach equilibrium and that process would stop. Well, that's where the electron transport chain, that third part of cellular respiration, comes into play. The electron transport chain is a collection of molecules in that inner mitochondrial membrane. So we are still in that same place. This right here is the mitochondrial matrix, that very inner section of the mitochondria that has a low hydrogen concentration. And outside of here, the outer part of this diagram is the intermembrane space. And I'm going to abbreviate that so you don't need to watch me write that whole word. But this is that intermembrane space. So we're still in the mitochondria, but we're in that inner membrane. And right here, in fact, is our ATP synthase, that final machine that's going to help make the ATP. But next to ATP synthase, we have this whole sequence of other molecules in that same membrane. And the thing about these molecules, they're made up of several different types. They're mostly different proteins called cytochromes. There are also some called ubiquinone. That's one you'll hear sometimes. That's this Q, and cytochrome C. They can move around a little bit within the membrane. But the important thing here is that each of these molecules, each of these protein complexes, is more electronegative than the one before it. So this is that electronegativity hill that we were talking about. The electrons that arrive here in this NADH, remember the H brings the electron. This electron is more attracted to this first protein complex than it was to this hydrogen. So it'll leave this hydrogen and bond to this. That lowers its energy, it releases some energy, and that energy is used to pump hydrogen ions out through that complex. You can see there's hydrogen ions moving out. But then this complex number 2 is more electronegative than the first. So that electron, once it's here, all of a sudden it wants to move here and it releases a little more energy. And then it wants to move here and it releases some more. And then it wants to move here. Each one is more and more electronegative. So the electrons travel through here. And as they fall down that hill, they release energy. And that energy is used to pump hydrogen ions out into this intermembrane space. And that's what increases the concentration of hydrogen ions out here. At the very end, this electron is finally donated to an oxygen molecule. So you should write in oxygen at the very end. Oxygen is the final electron acceptor. Remember, oxygen is like the most electronegative thing out there in terms of biology. So oxygen is what finally picks up those electrons, bonds with a couple of hydrogens, and eventually becomes H2O, which is one of the products of cellular respiration. We end up with some, wait, did I just say that wrong? Nope, that's right. It bonds with some hydrogens to form water. Sorry, I had to think all the way through that. So this transporting of electrons down this electronegativity gradient, that energy is used to pump protons out here. And then remember, those protons diffuse back through ATP synthase, which then creates ATP and makes that energy at the end here. So that's where that proton motive force comes from, P. So the energy from this electron transport chain here pumps hydrogen ions out to create the proton motive force, and the diffusion of those hydrogens back in powers the synthesis of ATP. This process of using a hydrogen gradient to do work is called chemiosmosis. So osmosis refers to the diffusion of those ions, chemi refers to chemical, and that energy of chemiosmosis is used to drive cellular work. Here, that work is ATP synthesis by a process called oxidative phosphorylation. That's a really fancy word, but if you look at it, oxidative just means there's a bunch of redox reactions. Remember, as electrons are transferred from one to the next down here, those are redox reactions because we're moving electrons. Phosphorylation just means adding phosphate, and the way you make ATP is you take ADP and you add phosphate to it. Okay, so oxidative phosphorylation just means adding a phosphate through redox reactions. So this is another example of energy coupling. We are using diffusion to power another process. And there are variations of this also used in chloroplasts. So you'll see this again in photosynthesis. And prokaryotes, bacteria, use this as well. And we'll talk a little bit about that towards the end of the week. So adding all the vocab in here, electron transport generates the proton motive force that's used by ATP synthase to make ATP. So where did the electrons for that electron transport chain come from? Well, they were carried here by that NADH and FADH2 that came out of these first two steps. Glycolysis is the very first step, which occurs in the cytosol. This word kind of tells you exactly what this means. Lysis, the second half of glycolysis, to lyse something means to break it, like a lysosome breaks things down. To lyse something is to break it, and here we're breaking glucose, so glycolysis. It takes glucose and splits it into 2 3-carbon sugars called pyruvate. In the process, it releases a little bit of ATP and also transfers electrons onto that NADH, that electron shuttle. Then the next step is called the citric acid cycle. Before this begins, this pyruvate needs to be converted to a molecule called acetyl-CoA. And then that acetyl-CoA enters the citric acid cycle. Depending on the source you look at, this used to be called the Krebs cycle. Don't ask me why they changed the name, but you will see both names. It's the same thing. This is a series of chemical reactions that you do not need to know all the details of. You need to know that it produces a little bit of ATP, but it also fills up more of these electron carriers. It adds those electrons onto NADH and FADH2. So all these electron carriers from glycolysis and the citric acid cycle bring those electrons over to that final step where they can be used in that electron transport chain to pump hydrogen to power the synthesis of ATP. So back to our overview to kind of put this all together, glycolysis and the citric acid cycle supply those electrons through redox reactions. They transfer those electrons onto here. That's a redox reaction. They do produce some ATP through a slightly different process called substrate level phosphorylation. It's not very much. There's only about 2 molecules of ATP that come out of each step here. Most of the energy goes into these electron carriers, and those electrons then fall through the electron transport chain, which powers the pumping out of protons to create that proton gradient. And then as those protons diffuse back into the mitochondrial matrix, that gives energy to ATP synthase to regenerate and produce the ATP. This final step, that process of oxidative phosphorylation, makes like 30 ATP molecules compared to 2 here and 2 here. So the vast majority of energy is made at this very last step. So here's another way to kind of visualize this. The energy in here starts, of course, in the bonds of a molecule of glucose. That glucose passes through the cell membrane. It gets split in half through glycolysis. Here's your first step out in the cytosol, which does produce a little bit of ATP. It produces 2 pyruvate molecules, that's that half glucose, but remember, it also creates some of those electron carriers, that NADH, you can add that in here. It fills up a few of those, which carry their electrons into the electron transport chain, so those are going to come back here. The pyruvate, so the energy is really transferred into here and here. The pyruvate then moves into the mitochondria and the matrix, where it is broken down even more, which produces a little more ATP. But again, it also fills up a bunch more of those electron carriers. So the energy here really goes from glucose into these electron carriers, both here and here, which bring it to the electron transport chain, where the energy moves down this chain with these electrons. And that energy is used to pump protons out into this space. This is that intermembrane space, so you can put some pluses in there if you've got room. And then as those protons diffuse back through ATP synthase, which would be right about here in our membrane, that energy is used to create ATP, which is our final goal of this process. Like I said, it happens in many, many steps so that that energy can be released in a controlled, usable way by your cells. So I know that's a pretty complicated process. This is one where you might need to watch this video a couple times to make sense of it. I would strongly recommend that you draw yourself some diagrams. Pictures are really helpful in trying to figure out this process. We're going to continue working with this process throughout the rest of the week to review and put this together. So I hope you have a great rest of your afternoon. Continue to work on your Mastering Biology homework. That will also go through these steps and help explain that to you. Let me know if you have any questions, and I will see you tomorrow.